As we all know, the most advancing area nowadays in nuclear molecular imaging includes the following areas: receptor imaging, radioimaging, gene imaging, apoptosis imaging, metabolism imaging. And among them, the receptor radioimaging is the most advanced, and some already applied in clinic. So in part 3, I will give a general introduction about receptor radioimaging. Characteristic of receptor imaging. First, we should know what is receptor. Receptors are the structures, usually proteins, on cellular membrane that after interaction with specific ligand, usually called first messenger transmitters, trigger a signal causing defined response mediated by the secondary messengers G protein coupled receptor or ion channels, ligand-gated ion channels. Just like this photo, receptors like a lock and a ligand like a key. One key can open one lock. Receptors can be characterized by their affinity and density. Affinity means binding between receptor and ligand slow or quickly. Density means the numbers of the receptor, or we call the quantity of the receptors. Classification of receptor. First, cell surface receptors. Ion channel linked receptor, G protein linked receptor, enzyme linked receptor. And second, receptors in nucleus, such as some hormones receptors in nucleus. The relationship between receptors and disease. Receptors mediate many vital communications between extrinsic chemicals and the nervous muscular system. Receptors also mediate some communication between different organs in the human body via endocrine system. Receptors mediate the communication between cells and also within the cell itself through the environment via the autocrine system. Basic characteristics of the receptor ligand binding. First, satuability. Because of the number of receptors in each cell is constant. For example, on the surface of mast cell, there were 8,000 receptors of Fc receptors of IgE. The ligand receptor binding reaction curve can be saturated. The second, specificity and high affinity. One kind of receptor only reacts with a certain structure ligand and has a stereo structure specificity, high affinity, and a low binding capacity. The third, reversibility. Receptor bind to ligand to form a receptor-ligand complex, and this complex can be divided into two parts, receptor and ligand. This process is reversible, both in vitro and in vivo. The fourth, consistency with physiological effect. The specific binding of the receptor and ligand ensures high recognition ability of receptor to tens of thousands of bioactive substances in vivo, and match the physiological and pharmacological effects caused by the ligand. Importance of the receptor imaging. Receptor imaging provides a unique and non-invasive means for studying the distribution, localization, quantity, density, and function affinity of the human receptors and physiological conditions. Neural receptor imaging has become an important tool for the diagnosis and research of some neuropsychiatric diseases such as Parkinson's disease. And this dopamine D2 receptor imaging of the healthy people, and this one is dopamine D2 receptor imaging of Parkinson's disease patients. Next, I will talk more about nuclear medicine receptor imaging. First definition. Receptor radioimaging is the introduction of the radionuclide labeled ligand into the body in vivo. Based on the principle that radionuclide labeled ligand can bind to specific target receptors with a high affinity, SPECT and PET are used in vitro to display their receptor binding sites at specific sites, and the functional metabolic image of the quantitative receptors and their spatial distribution of receptors reflected by their density and affinity of receptor respectively. Early detailed receptor imaging studies were mostly in the study of human brain disease. The first human receptor imaging was reported on the dopamine receptor using positron emission tomography by using carbon-11 labeled 3N methylspaferin NMSB. It was found that the D2 receptors are elevated in patients with schizophrenia and depression. Subsequent development in the single photon emission chromatography (SPECT) tracer led to the use of 1,2,3-iodine labeled iobenzamide for SPECT imaging of human D2 receptor. Key points for ligand in receptor imaging. Ligand should with high affinity and specificity to a specific receptors. 2. Ligand can obtain the high biological activity after radionuclide labeling, both in vitro and in vivo. Radiolabeled ligand is introduced into the body through appreciate delivery pathway, binding a specific receptor, binding in vivo. SPECT and PET were used to display receptor distribution and density. Radioligand. The choice of receptor system should originally based on in vitro binding affinity and specificity data. In general, because of the high specific activity available with the radioactive ligand and the concern of the excessive dosimetry of humans, KD as an indicator of the affinity about the ligand to the receptor should be in the range of 0.1 to 50 nanomole. When the affinity is too high, the radioactivity determined in the target organ is independent of the receptor concentration. The optimal condition for the change in binding detection occurs when the ligand-receptor ratio is suitable. Too much or too little tracer ligand in the vascular culture will decrease the ability to detect significant alteration. Equipment of receptor radioimaging, PET, positron emission tomography, represents the most selective and sensitive method for measuring receptor density and interaction in vivo. SPECT, single photon emission tomography. It can be applied, but detector efficiency is much lower than PET. It will provide less quantitative accuracy than PET. Radionuclide commonly used in receptor imaging For SPECT, most of ligand applied in human were labeled with iodine 123. Half-life is 13.2 hours, 159 keV. Technetium 99M, half-life 6 hours. The energy is 140 keV. For PET, the ligand applied in human were usually labeled with carbon-11, half-life 20 minutes, or F18, half-life 110 minutes. They are both positron decay radionuclide. From this table, you may see those radiopharmaceutics already used in clinic. Targeting receptors, example, for dopamine transporter receptors, for 5-HT, for monoamino oxidase A, for gamma aminobutyric receptor, gamma GABA, mainly labeled with positron decay radionuclide, carbon-11 or F18. Okay, that's all for this part. Thank you.